here to learning objective number 2, describe the chemical messengers and receptor types associated with the peripheral nervous system. So we began looking at sort of the general anatomy of the peripheral nervous system. We talked about the two branches, the rest and digest being the parasympathetic side, the fight, flight, freeze being the sympathetic side. And then we are now going to speak about how those signals are relayed. What are the chemical messengers? What are the receptors that are responsible for relaying these types of signals? So the types of cholinergic receptors are going to be receptors that bind acetylcholine. And then the adrenergic receptors are going to be receptors that bind norepinephrine. Now, we want to think about norepinephrine in two forms. It can be a neurotransmitter at the synaptic junction. So if we're going from one neuron to another neuron, norepinephrine is released as a neurotransmitter in that scenario. But also, because we have that third route of sympathetic communication, where a preganglionic sympathetic neuron can synapse onto a chromaffin cell that is in the adrenal medulla gland, and then the adrenal medulla chromaffin cells can release norepinephrine in the form of a hormone. We also have to think about norepinephrine being circulated in the blood in this form as well. So when we speak about norepinephrine, we think about it as a neurotransmitter, but also as a hormone in the case of that third route of communication. All right. Now, if we think about the actual neurotransmitters, so preganglionic neurons always release acetylcholine. That's a rule that you're going to want to commit to memory. Anytime we have a preganglionic neuron, the only neurotransmitter that's released is acetylcholine. Now, when we think about postganglionic neurons, which is the second neuron in series, there are some different options depending on what division of the nervous system we're thinking about, parasympathetic versus sympathetic. It also depends on specifically what tissue location of the body, what effector organ are we looking at specifically. So for the parasympathetic postganglionic neurons, they will also release acetylcholine. For the sympathetic postganglionic neurons, they will usually release norepinephrine, but a small exception exists for the postganglionic sympathetic neurons that will go to sweat glands. They will be the exception to the rule, and they will secrete acetylcholine. But for the most part, sympathetic postganglionic neurons will usually release norepinephrine as their main neurotransmitter. And then again, we have to include the adrenal medulla chromaffin cells, again, because they are releasing these as hormones. They're going to release 80% epinephrine, 20% norepinephrine, and norepinephrine can also be converted to epinephrine in the periphery, so that's really going to have somewhat of the same effect. And so the adrenal medulla will also release norepinephrine in the form of a hormone and not so much a neurotransmitter. All right, so let's illustrate that in terms of looking at the rules. So the rule is that all preganglionic neurons always release acetylcholine. And we can see that here, right? Here's our options here for the sympathetic nervous system. Our three options or three routes of communication. One is to a ganglia that is a part of the sympathetic chain. Here is to the adrenal medulla chromaffin cells. And then here is to a ganglia that is a collateral ganglia, right? Not a part of the sympathetic chain. So I'm actually going to write that in. So the sympathetic chain is our first option, our chromaffin cells, second option, and then a collateral ganglia. Hope I spelled that right, collateral ganglia, being the third option. So as we can see, all of the preganglionic neurons, all of them will release acetylcholine as their neurotransmitter. So that's a rule that you can kind of stick to pretty much memorizing as far as all preganglionic neurons. For the postganglionic neurons, right, the second neuron, excuse me, these are going to depend on the effector organ in question as well as sympathetic versus parasympathetic. So because we're looking at the sympathetic system first to start, the postganglionic neurons here will usually release norepinephrine. All right, usually release norepinephrine. For the chromaffin cells, it's going to be both. It's going to be norepinephrine plus epinephrine. Okay, and then we also want to think about the exception. So for sweat glands, postganglionic neurons that go specifically to sweat glands, these are going to have the exception to the rule where these guys will secrete acetylcholine. Okay, so this is the sympathetic nervous system, and this is kind of how what neurotransmitters slash hormones are released from each neuron in question. Let's switch gears and look at the parasympathetic side. So once again, we have this much longer preganglionic neuron. We have a ganglia that's far out and closer to the effector tissue in question. And as we can see, that rule still applies here. All preganglionic neurons will release acetylcholine, but then our postganglionic neuron here will also release acetylcholine. So another rule that we can apply is that all parasympathetic neurons will release acetylcholine. Okay, and so we can think about the rules that apply as far as things that are always the case, and then we can think about some of the exceptions that apply as well. So let's think about our receptors now. So we talked about the neurotransmitter slash hormone that's released from these neurons. Let's think about the types of receptors that have to be in place for these neurotransmitters to bind in order to relay that synaptic signal. So the nicotinic cholinergic receptors, these are going to be examples of ionotropic receptors, and we talked about these being fast receptors previously. And so the same principle applies here. Because these are fast receptors, the receptors also behaving like a channel. As soon as that acetylcholine binds to this nicotinic receptor, it opens up the channel, ions begin to move, that results in depolarization, and then we have the signal being relayed from the presynaptic neuron, or preganglionic neuron in this case, to the postganglionic neuron. So that's one example of a cholinergic receptor. So both of these are cholinergic receptors, the nicotinic and the muscarinic. So they both will bind acetylcholine. The only difference is the nicotinic is a fast receptor. The muscarinic, on the other end, is a metabotropic receptor. And remember, we described that as being a slow receptor. So what this means is that when the acetylcholine binds to a muscarinic receptor, it's not going to be a fast, immediate response the way that we see for nicotinic receptors. It's going to be slower. It's going to require some second messengers involved. It's going to require the entire process of signal transduction, where we have a G protein that's coupled to this muscarinic receptor. We have a part of that G protein dissociating off and having some effect downstream that will eventually result in an effect on the target cell. So we can already begin to see that although both of these receptors bind acetylcholine, although they're both classed together as being cholinergic, they have different mechanisms, they have different speed of response, depending on whether it's a nicotinic or it's a muscarinic type of cholinergic receptor. Okay, let's look at the location of these cholinergic receptors. So the areas where we find them will differ as well. So on the autonomic postganglionic neuron, on the soma of that neuron, we're going to find nicotinic cholinergic receptors. On the effector organ in the parasympathetic side, we're going to find muscarinic cholinergic receptors. All right. We're also going to find nicotinic receptors on skeletal muscle. So we'll talk about this entirely differently when we talk about the somatic nervous system. All right. So we also want to think about these receptors being there, although we're not really looking more specifically at the somatic nervous system at this time. And then again, our exception to the rule is that postganglionic neurons on the sympathetic side will have acetylcholine being released, Therefore, sweat glands on the sympathetic side will also need to have a cholinergic receptor, and that will be muscarinic cholinergic receptor. Okay. I want to go back here and just illustrate this a little bit better. So let's look at where these receptors are. So let's look at the sympathetic nervous system. And we can see that on the, let's go to the parasympathetic. On the effector tissue for the, sorry, I'm having a brain fart here. Let's go back here. So this is what I want to begin with. So looking at the postganglionic neuron. So here's our preganglionic neuron, here's our postganglionic neuron. Notice on the soma of the postganglionic neuron, we have nicotinic cholinergic receptors. And so this is going to be a fast response. This needs to be a fast response. If we consider on the collateral ganglia, there are also nicotinic cholinergic receptors also having a fast response. So as soon as acetylcholine is released from our preganglionic neuron, it binds to this fast nicotinic receptor, it opens up a channel, and it immediately depolarizes the postganglionic neuron, and then action potentials can move down towards the postganglionic neuron. So I say this to say that the location of the fast receptors, if we kind of zoom out, this makes sense. So this is not going to be a tissue-specific response. This is not going to need any second messenger. This doesn't have to have a response that is localized, depending on what effector organ is being innervated. This is an immediate response. As soon as acetylcholine binds to nicotinic receptors, it's going to automatically result in depolarization so that the postganglionic neuron can now send that signal all the way down to the effector tissue in question. And now when we get to the effector tissue, the receptor here, so in this case, it's going to be an adrenergic receptor. The receptor here will then have to be a metabotropic receptor. It's going to have a tissue-specific response. Depending on where this tissue is, it's going to result in some different type of amplifier enzyme, some different type of second messenger, and a whole different cascade of signal transduction events, depending on exactly what type of effector organ we are dealing with. So this needs to be a slow and more specific response. Here, this doesn't need to be a slow and specific response. It just needs to depolarize that second neuron. And so here we can have fast nicotinic receptors to get that signal quickly from the preganglionic neuron to the second postganglionic neuron. All right, and same is true for the parasympathetic side. On the postganglionic neuron, we have these fast nicotinic receptors. When we get to the effector tissue in question, we have a muscarinic receptor. Although it's also cholinergic, this is still going to be a slow receptor, again, because we have a more tissue-specific slow response taking place on the effector tissue to bring about the effect that is required for that tissue. Right. Let's look at the adrenergic receptors here. So we talked about the cholinergic receptors. We had two types, nicotinic being fast, muscarinic being slow. When we look at the adrenergic receptors, they're all slow. There is no fast adrenergic receptor. They are all coupled to G proteins. They're going to be two main types, and then further having subtypes, alpha and beta. The alpha receptors are going to have alpha 1, alpha 2, and beta receptors will have beta 1, beta 2, and beta 3. And now we want to think about these receptors as being able to bind norepinephrine and epinephrine, but it's going to have a different effect depending on where we have these receptors. And we're going to find different receptors on different types of tissues, which is what we're going to look at next. So if we think about alpha adrenergic receptors, they're usually located on things like blood vessels, the smooth muscles of our blood vessels. They're going to be a part of the sympathetic nervous system only. So adrenergic receptors are in no way related to the parasympathetic system. They're going to be the most common and most abundant throughout the body, and they're usually going to be excitatory. And when we say excitatory, this means that they're going to increase their tissue response. All right, I really want to make the distinction here because this isn't often a point of confusion in terms of what they do to their second messenger versus what they do to the tissue in terms of that effect that they bring about. So excitatory is usually referring to the tissue effect. All right, the effect that they have on their tissue in question. The affinity will have, or they will have greater affinity for norepinephrine over epinephrine. So they're going to more favor binding norepinephrine than they will epinephrine. Beta receptors generally will activate their cyclic AMP messenger, and then they will typically have differing affinities. So we're going to see that beta 1 has a different affinity, beta 2 has a different affinity, etc. For alpha receptors, they all prefer norepinephrine over epinephrine. Now let's look at the individual location of beta receptors, because these are kind of a little bit more specific. So beta 1 receptors will be on the cardiac muscle and the kidneys. They're going to usually be excitatory. And again, excitatory here means what they're going to do to their tissue, so their tissue effect. They're going to have equal or even affinity for norepinephrine and epinephrine. So they're going to equally prefer both of these. And then they're going to be, again, yeah, we said cardiac muscle and kidneys. Beta 2 receptors on the other end are going to be in blood vessels and smooth muscle. Beta 2 receptors are also going to be in the lungs, right? This will add here the lungs as well as the uterine. Smooth muscle are 2 additional areas where we find beta 2 receptors. And then these are usually inhibitory to their effector tissue. So again, when we talk about inhibitory, they're going to decrease activity on the effector tissue, or they're going to decrease stimulation of their effector tissue. That's another way to say that. Whereas beta 1 is going to excite or increase the excitation or sensitivity or stimulation on their effector tissue. Beta 2 receptors, on the other hand, are going to have greater affinity for epinephrine over norepinephrine. Lastly, beta 3, usually found on adipose tissue, usually excitatory to its effector tissue, and then this will also have equal or even affinity for norepinephrine and epinephrine. Okay. So these are our main receptors and sort of the principles that apply. What we're going to go on to look at is the mechanism of these receptors.